What are different NGS methods? So different NGS methods cover that what data we want to process. For example, do we have the DNA data, or we have the RNA sequences, or we have the protein data from the patients or the samples. So there are different type of technologies. For example, when we want to do DNA sequencing, so there we talk about genomics, and that will cover metagenomics, target sequencing, whole genome sequencing. Next, when we talk about RNA sequencing, so we talk about transcriptomic quantification, small RNA profiling, and we talk about protein sequences, and that will include ribosome profiling and epigenetics. Next is, what are the common approaches within NGS and how we generally process or do sequencing. So there are three major common approaches. First include genome sequencing, where we talk about the whole genome of the species. If we talk about the human, just for an example, then we take the whole genome of a human and then we sequence that genome to determine where are the mutations and what are the variants on that genome. If we are doing research on some disease, so we are interested to identify that what variants are different within the disease samples and the normal samples. Next is exome sequencing. So when we talk about exome sequencing, we are interested in just the protein coding sequence, because that protein coding sequences are majorly responsible in diseases. When we are interested in just the protein coding sequences, so we do exome sequencing. And when we are interested in a specific gene, for example, there is a person with any disease, for example cancer, so we are interested in what genes are mutated or differently expressing in that person. Specifically, when we talk about personalized medicine or personalized treatment when we are given to some person, so we are interested that which gene is specifically differently expressing or mutated in this person, so that or we can use some therapeutic approaches on that person. So in that case, we make targeted gene panel, and then we filter out specific genes in which we are interested. For that gene only we do the sequencing, because in that case we will be knowing that on which chromosome that gene exists. So for that we will do specific gene sequencing. Next is application of next generation sequencing in different spheres of life, where we are interested to identify different findings. So first will be the epigenetics, where we talk about the chronological age of a person, identification of different tissues, what are the habitual behaviors. So all these things we can determine with using epigenetics. We are interested in microRNAs, we are interested in phenotype, like physical appearance of the person, that different skin color, different hair color, different eye color, age, hair shape. So these kind of things we can perform using NGS sequencing. And then human identification, like how two people resembles with each other, what is their relationship, so we can identify, because sometimes we have observed that two cousins have similar nature of doing things and something like that, so we can identify with that. Then non-human identification also we can do, like other than humans we can sequence the animals, plants and other genomes as well. Then based on the ethnic differences, and one person lives in the eastern countries, one person lives in western countries, so there may be difference in the mutation and the gene expressions of that person to the other person. So next is basic NGS analysis workflow. If we try to understand, then first step will be nucleic acid extraction. So the sequences on the DNA, we try to first extract from the samples that we have collected from the patient. And if we are interested in RNA samples, RNA sequencing or DNA sequencing, so based on that we will perform processes to extract those samples, and we move to next step, which is library preparation step. So in this step we try to add the adapters to those sequences which we have like extracted from the samples, so that it will move to the next step for sequencing. So library preparation is very important step before sequencing. Basically adapters are the flags that we add on both ends. Here we can see the pink ends where we have added the adapters, so which will flag that this specific sequence is coming from which sample. And then the sequencing step will come where we will provide this particular library prepared sample to the sequencer, and sequencer will give us output in different file formats which we will process further to determine the variants in the sample. Next is